I'm Ken Binmoeller. I'm going to kick off this session on GI innovation, speaking on the creation of LAMS. It's been at times a very challenging journey, but always exciting and in hindsight, very gratifying. My thanks to Benjamin and Drew for the invitation. Now, Drew provided this title, not just out of the box, but through the box. Maybe I'll make a small edit. How about through the circle? This is the logo for Xlumina, the company that I created in 2008 to develop the LAMS. And as you can see, we have a device that is traversing this circle, and we're going through the circle transluminal across lumens, Xlumina. I have no financial conflict of interest. Drew provided a list of questions that he wanted me to address in the talk. And the first is, what clinical need triggered the initial thoughts about the invention? So I thought back. Where the journey started was in 1992. I was very privileged to work with Nib Sahendra in Hamburg, Germany, and we performed the first EUS-guided drainage of a non-bulging pseudocyst. We used a prototype curved linear array echoendoscope from Pentax that had a 2mm channel. We punctured the cyst with a homemade device that allowed us to immediately pass a wire into the cyst. This needle had cautery. We then exchanged the echoendoscope with only a 2mm channel for a 4.2mm duodenoscope over the wire. We dilated the tract and inserted a 10 French pigtail stent for drainage. And here you can see the needle traversing under direct EUS guidance from the stomach into the cyst. So this was the first EUS-guided application of the Seldinger technique, named after Sven Seldinger, the radiologist. The over-the-wire technique consisting of 4 steps. First, accessing the target cavity, usually an FNA needle, inserting a wire through that needle, then dilating the tract to prime it for placement of a stent to drain the target cavity. As elegant as the Seldinger technique is, it is a multi-step technique. And with each of these steps, we take risk. So it's a multi-risk procedure. Needle puncture, wire through the needle. When we remove that needle, there's a gap between the wire and the tract, and we can have leak. When we insert the dilator over the wire, we may displace the target cavity from the bowel wall. When we dilate the tract over the wire, this can lead to perforation. And when we remove the dilator, this again can result in leak, that gap between our guide wire and the tract. And when we insert the stent for drainage, this once again can displace the cavity from the bowel wall. This video illustrates our most dreaded complication, perforation, occurring during pseudocyst drainage using the Seldinger technique. And you'll see the stent from the stomach lumen after placement. And I initially thought that the stent was in the cyst, draining the cyst. But as I looked more closely, you can see fat. This is mesenteric fat in the peritoneal cavity. So the stent had in fact been inserted alongside the cavity and not into the cavity. And this patient ended up having to go to a surgery. When we perform EUS-guided transmural drainage, we are, in fact, creating an intentional perforation of 2 walls. So we're creating 2 holes, one in the bowel wall and the other in our target cavity. And if the target cavity is not adherent to the bowel wall, then we risk leak with complications. And the best example of a non-adherent target cavity is a gallbladder. And if you look with EUS, you see this very well-defined echogenic layer interposed between the gallbladder and the duodenum. Now, it may look like the gallbladder is adherent to the duodenal wall because it's pushing up against the gallbladder wall, but as soon as you start to drain this gallbladder cavity, the gallbladder will displace from the duodenal wall. So what was the clinical need? It was for dedicated EUS-guided tools to prevent leak. We were using off-the-shelf tools that were designed for ERCP, and we managed, but these had shortcomings. We needed a transluminal stent that was covered, fully covered and self-expanding to seal off the tract so there isn't leak. Should be lumen-apposing, and it should provide a robust port for transluminal intervention so we could drive our scope through the transluminal stent. And we needed an exchange-free delivery system that eliminates over-the-wire exchange. So in essence, eliminating the Seldinger technique. We could access the target lumen with a stent-loaded catheter, so we don't provide any opportunity for leak to occur with an over-the-wire exchange. I patented the LAMS concept over a decade ago in 2009, and the patent was issued in 2013. So it took a number of years. The abstract summarizes the concept as a device whereby two luminal structures in the body may be drawn toward each other and a fluid conduit formed in between. This concept has three major components: anchoring lumen-apposing flanges, a self-expanding conduit saddle between the flanges, and it's fully covered. Now, what about the name, Axios? I like names that capture the essence, the spirit of what the device is supposed to serve. And what we are creating with the LAMS is an axial ostomy. We are creating a connection between two lumens to enable flow between those two lumens. And so axial ostomy was abbreviated to Axios. And it so happens that Axios is the Greek mythology river god. So very appropriate for this device. And I founded the company Ex Lumina, the Across Lumens company in Mountain View, to go beyond with advanced transluminal therapy. And here is the team that I pulled together after founding the company, and our CEO here, Michael Allen, and our engineers. Some in the audience might recognize Tom DeSimio, who was my nurse at CPMC for over 10 years. And he served as a trainer and my sidekick performing all of the animal studies. So the first challenge, and it's a big one, is fundraising. And we raised $6 million in our Series A. It took a few years to raise the funds, and we received the funds in 2008. Drew's second question was, what was the evolution of the innovation from the initial concept through various ideas and prototypes? Well, you start with a drawing on the napkin with the concept. That's the patent that I showed you earlier. And now we have to translate that concept into an actual device. And this is where the benchtop prototyping starts. And that's what you need a lot of money for. Here you can see the workroom where we did the prototyping at Lumina. We define a goal, we identify the challenges, and then we find solutions to address those challenges. So our first goal, lumen-apposition. Our challenge was to avoid ischemia, and our solution was to use flanges with a very large surface. Our goal was to resist migration. Our challenge was to make the LAMS removable. Our solution was to use a double-walled construction that gave it robust pull-out strength, but still enabled removal. Our goal was that the LAMS should be compressible and yet resume its original shape after deployment. The solution was to use a proprietary wire braid. We went through countless prototyping studies to find just the right braid and diameter of wire and wire material, etc. The goal was uniform and durable coating. The challenge was that this should be sufficiently thin to enable insertion through the working channel, and it should be conforming. The solution was to manually brush the coating onto the LAMS. Prototyping the LAMS certainly had its challenges, but what I found most challenging was developing the exchange-free delivery system. And the concept that I came up with first, I called the multi-layered dog bone. I actually filed the concept back in 2006, and the patent was issued in 2013. This cartoon illustrates how the multi-layered dog bone concept works. It consists of 3 layers. The first layer is the FNA needle to puncture the cyst. And then the second layer is a balloon catheter with a dog bone configuration. And the third layer is the sheath with the LAMS mounted on it. So here in the cartoon, you'll see first the FNA needle extrude. You can pass, of course, a wire through that needle. This is the second layer. This is the balloon catheter. And when we inflate that balloon, you see how it takes on a dog bone configuration with 2 barbells here at the end. And the third layer now is the LAMS sheath. And when we inflate the balloon, this inflates the LAMS in a barbell configuration. And now we can remove the delivery system and leave the LAMS in place. Another concept that I came up with is the multi-layered switchblade. So instead of using an FNA needle, we're using a trocar. There's no lumen in this, with a switchblade at the end. It creates the 3.5 millimeter opening. And that enables us to then immediately insert and deploy the LAMS catheter. So here you can see the video showing how we enter into the gallbladder. You see the deployed switchblade. Now we inflate a balloon to snug this up against the gallbladder wall and keep it approximated in apposition with the bowel wall, duodenal wall. Then we deploy the LAMS and remove the entire device, delivery device. And here you see little stones that are draining out of the LAMS. And the third concept is the multi-layered cutter dilator. So here we are replacing the balloon catheter with a cage. So this serves as an anchor. And to penetrate through the walls, we are using a cutter blade that we deploy as we pass through the walls. And the outer sheath engages the anchor, as you can see here, and then we can immediately deploy the stent. The goal is always the same. It's to avoid any risk of leak during delivery of the LAMS. So these are just different concepts. We made prototypes of each of these, but finally, we decided to use what is now commercially available, the Electrocautery-enhanced delivery system. And so this has a ceramic bougie-shaped tip, and we have cautery micro wires alongside the conical tip, and then also converging at the very tip in a circular fashion, so that we are distributing the cautery over a very small surface area. So we maximize the cutting effect and minimize the coagulation effect. Now, the Electrocautery-enhanced delivery system was developed very late. And the reason for that is that I was resistant to using a cautery. My concern was that if we use cautery, and for any reason, the LAMS mal-deploys, then the hole that we've created will be less forgiving, is less likely to close after using cautery. So I was trying to find a method using cutting to avoid using cautery. Drew's next question was, what were some of the biggest challenges to making this a reality, and how did you overcome them? I could spend hours talking about this. There were countless challenges, at times extremely stressful challenges. In hindsight, I would say that the biggest challenge was regulatory, and that consumed a big part of the some 45 million that was invested in Nexlumina. LAMS was to be a PMA. I don't have time to go into an explanation of a PMA versus a 510K, but suffice it to say that a PMA usually requires a randomized controlled trial with a large number of patients to prove safety and efficacy. So it was initially determined by the FDA to be a Class 3 device. And we were very lucky, following multiple meetings with the FDA and countless hours justifying why this should be a Class 2 device. And in the end, the FDA agreed to making the LAMS a de novo 510K. And here you can see the announcement. FDA says pancreatic stents are lower risk at Nexlumina's request. We were able to convince the FDA that this is really no different than a pancreatic stent, inserted, of course, transpapillary, is just going transmural. And since pancreatic stents are Class 2, this too should be Class 2. And fortunately, the FDA in the end agreed, and in 2014, we received the de novo 510K approval. The FDA did require a registry to document rudimentary safety and efficacy, and it needed to be multi-center. So we funded a trial enrolling 33 patients at 7 centers, 6 US, 1 Europe. And we were able to demonstrate very high technical success and clinical success. And we had complications in 15%, but none of these were serious complications. So pain, stent migration, and dislodgement. Drew's next question, what was the aha moment when you realized this may actually work? So the first aha moment was during our animal studies, when we placed the final version of the LAMS, and we could demonstrate that it was robustly opposing the lumens, and we could drive a scope through this. And this is the gallbladder of the swine, and you can see the cystic duct filling and the bile duct draining. And we followed these animals over several weeks in survival studies, and the lumen remained patent. And then on necropsy, we could see fusion of the wall layers. So this led to the publication of our results. So this was really a huge aha moment that this actually can work. The second aha moment was the first in human, and we performed this in Japan since we couldn't do this in the US in 20 patients. So I worked with Takao at his center in Tokyo. Our next question, did you imagine it would have as big an impact as it has? Well, just look at the application of LAMS. I developed LAMS originally not to drain pseudocysts. It was to drain the gallbladder, and my goal was to eliminate or avoid cholecystectomy with transluminal drainage of the gallbladder. To get FDA clearance for gallbladder drainage, that would have definitely required a PMA, and it would have required a monumental work and funding. Now we're using LAMS off the shelf and off-label to treat luminal stenoses, be it pyloric or short strictures, for example, anastomotic. I'm going to end with 2 quotes from this great innovator who exemplifies thinking outside the box. The first quote is, "In the middle of difficulty lies opportunity." So challenges and difficulties during our procedures are welcome, but they should lead and trigger concepts for solutions. Second, if you always do what you always did, you will always get what you always got. So in our daily work in the trenches, we should all be thinking outside of the box or through the circle. Thank you.